we will introduce RDDC's AI tool, the RNA Splicer, as a crucial step in the regulation of gene expression. The precise regulatory mechanisms of RNA splicing have not yet been fully elucidated. In recent years, the impact of gene mutations on RNA splicing patterns and their molecular mechanisms has become a research hotspot and challenging issue in the field of biology. The tool introduced this time is developed based on AI algorithms and is mainly used to predict and analyze the impact of gene mutations on RNA splicing patterns and their potential regulatory mechanisms. First, our tool offers 4 flexible ways to input your data. You can directly paste standard mutation names, manually enter mutation details, or provide information based on protein or transcript information. Now, let's demonstrate the tool's functionality using a mutation in the USH2A gene. We'll choose the mutation method, which allows us to manually input the mutation details. First, type the gene name into the search box. As you type, the system will provide suggestions. Here, we'll select the human USH2A gene. The system will automatically display the most common transcripts for this gene, if needed. You can also select other specific transcripts. Next, we'll enter the specific details of the mutation site. Once you're finished, click on the blank space or press the Enter key, and the system will automatically parse the mutation site before and after the mutation. To better understand the mutation's location within the gene structure, you can click the Unfold Diagram button for a visual representation. After confirming the entered information, you can click the Add button on the left to include more mutation sites for prediction in the same analysis. Once you've reviewed and confirmed all the input information, simply click Submit. After submitting, you'll need to wait for a while for the AI model to perform the prediction. As you can see here, the model predicts that this specific mutation site may lead to 2 novel RNA splicing outcomes. To understand these prediction results in detail, you can click the question mark icon to access comprehensive documentation. Furthermore, if you're interested in exploring whether similar mutations in other species like mice and rats have the same impact, you can click here to analyze homologous mutations and their potential effects on RNA splicing. So, that concludes our introduction to the RNA Splicer prediction model. We hope you found this tutorial helpful. We encourage you to try out the tool and share your feedback. Thank you for watching.